The final speaker of today's session is Dr. Sliman Bensmaia from University of Chicago. I believe that Dr. Bensmaia is one of the most active researchers in the field of neuroscience of tactile texture perception. He's going to talk about the spatial and temporal mechanisms mediate the tactile perception of text. Dr.
Checkity check yourself. All right. So in my lab, I'm interested in neural coding. The general question that we're trying to address is neural coding. Let me see if I can. Which in our hands means that you have a stimulus, in our case, a spatiotemporal pattern of deformation of the skin, and it leads to a pattern of activation in the nerve. And so whatever information you have about an object grasped in the hand has to be conveyed in this pattern of spiking in the nerve. A couple of intervening synapses I'm not going to talk about today, the cuneate nucleus and the thalamus, but ultimately this activity in the nerve culminates in a pattern of neural activity in somatosensory cortex, the part of the brain that receives these signals from the hand, ultimately culminating in perception. And so what we're interested in is first understanding the relationship between the stimulus and the response. So understanding how the different stimulus properties are reflected in the neural response. But ultimately, we are also interested in understanding how these patterns of neural activation lead to perception. So when we grasp an object in the hand, we have information about its shape, its texture, a motion of this object across the skin. And somehow these patterns of neural activity, all this information is multiplexed in these patterns of neural activity. So we're trying to understand the relationship between stimulus and response and perception. So what we're going to talk about today is Shitsukan, I think, texture perception, and we are exquisitely sensitive. Sorry to the visual neuroscientists in the room, but we are actually much better at tactile texture perception than we are at visual texture perception. We are extremely sensitive to texture. We can distinguish textures that vary in element size on the order of tens of nanometers. We can distinguish textures that vary in terms of spatial period on the order of hundreds of nanometers. And somehow, so what I'm going to talk about today is what are the mechanisms that mediate these incredible abilities. So first I'm going to introduce the players. There are something like 13 different kinds of receptors in the skin, joints, and muscles, 3 of which are relevant to texture perception. Meissner corpuscles, Pacinian corpuscles, and Merkel discs. These are receptors that respond to non-noxious skin stimulation. Signals from Meissner's, and you know, they differ in a variety of ways. I'm not really going to talk about too much in terms of their functional properties, although I want to just point out that they're very, 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 very well understood. What drives Merkel, Pacinian, and Meissner corpuscles is well understood. Suffice it to say that they respond to different aspects of skin stimulation. And signals from Meissner's are carried to the brain with afferents called rapidly adapting or RA afferents. Signals from Pacinian corpuscles are carried to the brain with PC afferents, and Merkel's are innervated by slowly adapting type 1 afferents. So the activation of these 3 receptors conveys most of the information that is available about texture. Now, the sort of dominant theory of tactile texture perception is one that was developed by my postdoctoral advisor, Ken Johnson, and Ed. And here it goes. So in a very beautiful and elegant series of experiments in the 90s, it was shown that if you scan these stimuli across the skin, these embossed dot patterns, so they're like braille-like dot patterns, arrayed in regular patterns, like these, so these are the stimuli, and you reconstruct the response of entire populations of SA, RA, and PC afferents. This is what you get. I'm not going to spend a lot of time talking about how you do that, but just imagine you had godlike powers and you You can record simultaneously from every single essay in your finger. This is what that pattern of activation would look like. And then this is what RAs would look like, and this is what PCs would look like. And just looking at this, you can see that the spatial pattern of activation in essays closely matches or reflects the spatial configuration of the stimulus. This is somewhat true for RAs and not at all true for PCs. The PCs are kind of a mess. And this was interpreted as, well, as a first piece of evidence suggesting that essays convey information about texture. In that you had this very faithful representation of the stimulus in this population of afferents, and certainly much more faithful than that in RAs and certainly PC afferents. The second really compelling part of that, though, is, okay, so you have this nice spatial pattern of activation, but you have to sort of link it to perception, and so, this is just one of several experiments that showed, so the funny things about these patterns is kind of not really intuitive, is that as you increase the distance between dots on these patterns, the perceived roughness increases then decreases. So for whatever reason, when the dots are 3 millimeters apart, that's the roughest that these things feel. If the dots get further apart, it starts feeling smoother. If the dots are closer together, it starts feeling smoother. So that's what this dark, so this continuous line here shows is the ratings of roughness by these subjects, by human subjects, averaged over several human subjects, and you see this sort of upside-down U-shaped function. Now, what was striking is that you could predict very precisely how rough these things would feel based on this spatial pattern of activation evoked in essay 1 afferents. And I'm going to skip some of the math. I'll allude to it a little bit later. But basically, in order to predict the roughness, you could predict the roughness based on the spatial inhomogeneity in the response. So in other words, if all the essays responded the same or didn't respond at all, something would feel very smooth. And to the extent that some essays were active and some essays were not, something would feel rough. And that sort of makes sense because, so the idea is like a single Braille dot pattern is like poking into the skin and exciting an essay afferent, while other parts of the stimulus are not poking into the skin and therefore not affecting or exciting these afferents. And so it's this differential pattern of activation that signals the fact that there is some coarseness to the surface. And so this was a nice series of experiments that culminated in the conclusion that slowly adapting type 1 afferents convey information about texture in this spatial pattern of activation in a very vision-like way, right? So it's like this, the spatial configuration of the stimulus is reflected in the spatial pattern of activation, just like in the retina, and then presumably you analyze this spatial pattern very much like you would analyze a spatial pattern of activation in the retina. But remember I told you that we're extremely sensitive to texture, down to, in the nanometers, tens to hundreds of nanometers. The innervation density of the fingertip is on the order of millimeters. So if we relied on this mechanism, we would really suck at texture perception. And so we decided to do some experiments to further investigate the neural basis of texture perception. And our idea was that, look, if I tell you, name 3 textures, unless you're blind, Braille-like dot patterns isn't going to be one of them. You're going to say things like paper, various fabrics, whatever. And so we wanted to investigate tactile texture perception across the range of tangible textures. So we presented a bunch of kind of quote-unquote natural textures, 55 of them. And because these textures were not under experimental control, we needed a way to actually characterize their microstructure. So to that end, we used a laser microscope. To present the textures to human subjects and monkey subjects, we developed a texture robot that allows us to scan textures at a very precise contact force and speed across the skin. Ultimately, though, the receptors are embedded in the skin. They're not at the surface of the skin. So we thought to understand what the receptors experience, we had to understand how the skin responds to these textures. So to that end, we used a laser Doppler vibrometer that can really that very precisely measures how the skin is responding to these textures. Finally, we have a motor here, a vibration exciter, that allowed us, under certain circumstances, to very precisely stimulate the skin with these temporal patterns, these vibrations, because we can't really control how the skin responds to a texture. So we wanted to be able to control exactly what the skin did, and we did this in some experiments with this vibration exciter. On the neurophysiology, or on the data collection end, we brought 3 methods to bear on this problem. One, we recorded from peripheral nerve afferents. This is in a rhesus macaque preparation. This is actually an anesthetized rhesus macaque. Basically, we make a little incision, bear the nerve, dissect out individual fibers, wrap them around our electrode, identify the receptive field location and the type of fiber it is, which is trivially easy, and then we just scan all these stimuli across the receptive field. We also recorded from primary somatosensory cortex, the part of the cortex that receives input from the hand. This is in an awake behaving animal. These animals were doing just a visual discrimination test just to keep them awake. And then finally, because ultimately what we're interested in is perception, so we did some psychophysical experiments, and these are on human subjects. We brought to bear the incredible similarities between humans and macaques in somatosensation, so that we could, and what we wanted to do, again, is to understand how these recorded patterns of neural activation culminate in perception. So first I'm going to talk a little bit about our results in the nerve. How is information about texture conveyed through in patterns of activation in the nerve? So first, these are 4 textures, 4 example textures here. This is the rail leg dot patterns. We wanted to replicate previous results. Tactile is a coarse fabric, and these are 2 fine textures, fine fabrics. And these are the responses, the spatial pattern of activation is slowly adapting type 1 afferents. So replicating the result of Ken Johnson and colleagues, the spatial configuration, and we're representing these slightly differently, but it's basically the same type of display. This is average over large populations of SA1s instead of a single one. But basically what you can see is that the spatial configuration of the stimulus is reflected in the spatial configuration of the response, as had been found by Johnson and colleagues. And now you look at the response to hug towel, which is this coarse fabric, you see that the knots of hug towel are sort of represented in the response of these afferents, but sort of this like fine texture stuff doesn't seem to be there. And then when you look at the SA responses to nylon and chiffon, they're a complete mess. There's no discernible structure. So then, of course, we're just eyeballing things. So we did a little bit of more quantitative assessments of these various things. And what we found is, so this is the firing rate in SA afferents for 12 textures that range from fine to coarse. And you can see that these essays barely responded all to these fine textures, whereas they respond pretty robustly to things like braille-like dot patterns. Then we wanted to see, is there any spatial structure in the response? And what we found is there was no discernible spatial structure in the responses to fine textures, but there was strong spatial structure, which is obvious in the responses to coarse textures. And then finally, we wanted to take a correlation, because the idea was that there was an isomorphism between the response and the stimulus. We wanted to take a correlation between the response and the stimulus, and we found that this correlation was really high for coarse textures and basically 0 for fine textures. So these experiments showed that, yeah, for coarse textures, slowly adapting type 1 afferents can convey information. For fine textures, they're completely uninformative. So where does this information come from? How is it that we can distinguish silk from satin, good silk from cheap silk? Well, there are 2 other receptor types this is an example response from, or the responses of one example PC afferent to 3 fine textures. So these are raster plots. There's 42. This is the responses of one PC afferent to 42 representations of nylon, denim, and silk. And so these are the spiking responses here. These are the power spectra of the spiking responses. You can squint and see it in the spiking responses. You don't have to squint to see it in the power spectra. That there is a lot of temporal structure. So if you present nylon over and over again, you get this very periodic response in these PCs, and the frequency composition of that response is very consistent across repeated presentations of nylon. If you present denim, you're going to have a different periodicity, as you will for silk. So eyeballing this, it seems like these temporal patterns of activation in PCs could be very informative about texture. So then we did a little bit of math to try to assess the extent to which this was true. I'm going to skip a lot of the methodological details. All this stuff is published. It was published in PNAS last October. But what we did is just classification. The question was a very simple, kind of obvious question. Can you identify a texture based on the pattern of activation that it produces in each of these afferents individually at first, right? And so the idea is if two textures produce very different responses in an afferent, they'll be easily distinguishable from each other based on the responses of the afferent. And to the extent that these responses are overlapping, you're going to confuse these two textures. So that was the idea. The nice thing about this analysis, this was a metric space analysis, is that we can also vary the temporal resolution of the analysis. So the idea is that on one end, this end here, the infinity end, we're basically counting spikes, right? We're saying, we're going to say two textures are distinguishable from one another to the extent that they produce different numbers of spikes. On the other end, we are taking into consideration sub-millisecond differences in the spiking responses. And then, and so textures will be different to the extent that these sub-millisecond, or that their spiking responses differ in the sub-millisecond range, and then everything in between. And so to make a long story short, what we found was these nice functions, especially for PC afferents, where PC afferents were most informative about texture. So if you don't get anything of what I just said, hopefully you'll get the following sentence. The PCs were most informative about texture if you took spiking differences in timing into consideration at a resolution of about 2 milliseconds. The temporal resolution of RA signals was on the order of 5 milliseconds, and SA signals, basically, if you counted spikes, you were just as good as if you took the timing into consideration. So these temporal patterns are informative. So now you might be looking at this. This is proportion correct identification or classification as a function of temporal resolution. You're like, well, 25% isn't that great. The overall, the chance level is 1 over 55, so a little less than 2%. Still, though, 25% were way better than 25%. But keep in mind that this is with a single afferent. If we did this with 7 afferents, which is unfortunately how many PCs we had, we were basically at 100% across the board. And so this is proportion correct classification with a population of 7. Now, keep in mind, when you touch a texture, hundreds or thousands are responding. But with 7, you're basically at 100% across the board. So this is the textures broken down by quartile. So these are for the finest textures. These are the coarsest textures. You're very good across the board with PCs. You get better and better with RAs as the textures get coarser and coarser. And SAs really are pretty much suck at conveying information or texture information, except for the absolute coarsest textures. So anyway, so this showed that these temporal patterns of activation, particularly in PCs and RAs, are highly informative about texture. So in principle, could be used to mediate this ability that we have to distinguish different textures. So then, I'm going to talk briefly about this, but so then the question is, what shapes these temporal patterns? What determines the spiking response? And it turns out a very simple thing. The skin vibration. So I told you we had a laser Doppler vibrometer. We could actually measure the vibrations evoked in the skin when we scan these textures across the skin. And then we could look at the black here, the black trace, is the power spectrum of the vibrations evoked in the skin when we scan 3 different textures, chiffon, nylon, and taffeta. And the orange traces are the power spectrum of the evoked neural response in PC afferents. And across the board, you could really predict the PC response pretty well just based on the power spectrum of the vibrations, or the vibrations that were evoked in the skin. This was definitely true of PCs and to a lesser extent true of RAs. Okay, so now, here's a summary for those of you who are just waking up. A summary of what I've said so far. So coarse features are reflected in the spatial pattern of activation and slowly adapting type 1 afferents. Fine features are reflected in the responses in the temporal spiking patterns of RA and PC afferents. So the idea is, you know, like silk might go like DD, 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 and satin might go DDD, 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 DDD. They might evoke the same number of spikes, but they'll be distinguishable based on these different temporal patterns. At least in principle, right? But again, we're interested in perception, and we need to make a link between these neural, maybe there's some other thing that's conveying information about texture that we haven't thought of. So we wanted to make a link between these neural patterns and perception. And following tradition, really, we went back and looked at perceived roughness, which is like the dominant dimension in tactile texture perception. And so first, we went back and implemented the Johnson model, this idea that the spatial variation, the spatial inhomogeneity in slowly adapting type 1 afferents is what determines roughness, right? That's what's in the textbooks right now still. And what we found is, okay, we can account for 64% of the variance with based on SA1 spatial variation. And that's not bad, but that's not great, right? That's pretty good. That means there's some relationship there, but it's not the most compelling relationship. But I should also mention that to do this, we took exactly the algorithm that had been implemented before and had successfully predicted the roughness of Braille dot patterns, Braille-like dot patterns, and we applied that exact algorithm to see if we could predict the roughness of our textures, and we could only to a certain extent. So then we looked at the temporal variation, so the degree of temporal inhomogeneity in the responses of RA and PC afferents. So the idea is if an RA is just responding like did in a very regular way, something is going to feel much smoother than if it's responding in a temporally inhomogeneous way with fits and starts. And what we found is we do pretty well there. We can explain 88% of the variance with RA responses and 76% of the variance with PC responses. But there is no reason, you know, so you might be tempted to say, okay, well, I guess it's RAs. Johnson was wrong, it's RAs. Let's go home and have a beer. But in fact, why would you ignore these other highly informative signals? There's no reason that you would do that. So then what we did is tested the simplest perceptual hypothesis, which is that roughness is determined by a linear combination of these three, and suddenly we can explain all the variance in these perceptual judgments. Importantly, though, the coefficients of all three coefficients are significantly non-zero, which means that the perceived roughness is best predicted when you take all three signals into consideration. If one drops out, your ability to predict perception becomes worse. So this suggested that the perception of roughness relies on the responses of three afferent types, SA, RA, and PCs, and that it's determined by the spatial pattern of activation in SAs and the temporal pattern of activation in RAs and PCs. So then we wanted to see, okay, so now how are these signals interpreted in cortex? Well, there was already a pretty good theory out there of how these spatial patterns of activation are analyzed in primary somatosensory cortex, and it's in a very vision-like way, where you have, so this is stuff from Jim, this is stuff that we did later, where you have these neurons in primary somatosensory cortex, very much like neurons in primary visual cortex, that have excitatory parts in the receptive fields and inhibitory lobes in the receptive fields. So the idea is if you poke this part of the skin, you excite this neuron. If you poke this part of the skin, you're going to inhibit this neuron. If you poke both of these parts simultaneously, basically you're not going to change the response of the neuron much. And that's exactly the kind of computation you need to do in order to compute roughness based on these spatial patterns of activation. But of course, the next part, what we were interested in is to understand, well, what about the RA and SA parts? What about, RA and PC parts, so the parts that are related to the fine surface structure? How are those signals interpreted in primary somatosensory cortex? And for these experiments, we wanted to understand, so the idea, again, just to refresh your memory, the idea is that it's these complex vibrations that are produced in the skin during the scanning of texture that are transduced by SA and by RA and PC afferents and convey information about fine textures. So we were interested in then understanding how is it that these texture-like vibrations, by the way, the frequency of which ranges from 100 to about 500 hertz, how are these texture-like vibrations encoded in the brain? How does the somatosensory cortex construct representations of these sort of naturalistic texture vibrations? So to study this, we just used our vibration exciter. We stimulated the skin with a bunch of very well-controlled vibrations that varied from sinusoids to polyharmonic stimuli to more like texture-like bandpass stimuli, while recording the responses in primary somatosensory cortex, so the hand representation in primary somatosensory cortex, again in rhesus macaques. And this is what we found. And we were very surprised, and I sort of tortured one of my postdocs for a year. I was like, look, go find me a rate code. A year later, he could not find me a rate code, and instead, this is what he came up with. These are the responses to sinusoids. So this is the responses to 100, 200, 400, 800 hertz sinusoid. This is the spectrogram of the neural response of one neuron. This is the spectrogram of the neural response of this population of what we call phase-locked neurons. And what you can see is the frequency of the stimulus is represented in the spiking response of these neurons, up to 800 hertz. And this is made possible by very tight phase locking. So in the response to 100 hertz sinusoid, if the neuron spikes, it basically always spikes in the same part of the cycle, so you can decode the frequency of the stimulus from the phase locking of this neuron, up to 800 hertz. So this is a 1.125 millisecond cycle, and all or most of the spike happened within a third of that. And so you can decode the frequency from the timing of the response. So this, remember I was talking about how slowly adapting type 1 afferents and this whole spatial thing is very vision-like? Well, this does not look vision-like. This looks a lot more like audition-like, where you have this idea of phase locking to the stimulus to convey information. So of course, sinusoids are, kind of bogus stimuli because, textures are not sinusoids. So we thought, well, maybe this is an epiphenomenon. So then we looked at the responses to these more naturalistic vibrations. So this is the spectrogram of a patch of stimulus that lasted, 200 milliseconds. And so this is one of those bandpass stimuli. So the frequency composition of the stimulus is changing over time. And you can, if you squint, it seems like if there's a hot spot here, you're going to have a corresponding hot spot there, suggesting that the instantaneous frequency of the stimulus is represented. So this is the spectrogram of the stimulus, spectrogram of the response. It suggests that the instantaneous frequency of the stimulus is represented in the instantaneous spiking response, right? Or the frequency of the spiking response. This is squinting. We did a cross-correlation between these spectrograms and these spectrograms to see if this was a general phenomenon. And we found that indeed, from 50 hertz, which was the lowest frequency we tested, up to 800 hertz, there was significant cross-correlation between the spectrogram of the stimulus and the spectrogram of the response. And it was right there at the right delay, around 20 milliseconds. So not some kind of, we also subtracted from this cross-correlogram for you neuroscience nerds, we subtracted the cross-correlation that you would expect by chance, just by kind of using a rate-modulated Poisson process for the response as a control. So anyways, this is a lot of mumbo-jumbo. If you don't believe the cross-correlation thing, you'll believe this. These are, I think, illustrate the point I'm trying to make very clearly. If you have a sinusoidal sweep like, and you look at the neural response just follows it. So whatever the frequency of the stimulus is going to be reflected in the spiking response. So from this, we showed, and I'm skipping a bunch of things because this was really kind of went against the grain. So we did all kinds of different controls and for instance an information theoretic analysis that showed that you could not decode the frequency of the stimulus from the rate. You had to look at the timing, and also that the information in the timing was precisely corresponded to perceptual performance on frequency discrimination tasks. So you could predict the performance of human subjects in a frequency discrimination task based on the timing of the responses of the temporal patterning in these spiking responses. Of course, vibrations vary not just in frequency, they also vary in amplitude. And so we wanted to see how is information about amplitude encoded in the responses of these neurons. And what we found was very surprising to us, in that the firing rate, so this is the amplitude this is for sinusoids and for noise, but it's showing the same thing. This is the firing rate as a function of RMS amplitude, and the color scheme shows different frequencies, and all these stimuli just kind of cluster together. So the firing rate is completely independent of amplitude. And this is very surprising because if you plot this for peripheral afferents, particularly PC afferents, it flays out. The strength of the response is highly frequency dependent, and in cortex it becomes completely frequency independent. But that's a nice thing. It's not a good thing if you're trying to decode frequency from firing rate, but it's a nice thing if you're trying to decode amplitude from firing rate. So what we then did is we took our bandpass stimulus and we just computed the envelope of the stimulus, of the vibration, using a Hilbert transform, and that's the black trace. And then the gray is the population response of these neurons to these stimuli, and you can see that the waxing and waning of the stimulus is reflected in the waxing and waning of the response. And then we did, of course, a information theoretic analysis to show that if you want to, that information about the amplitude of the stimulus is contained in that spiking response, the strength of the response, and not in the timing of the response. So this was kind of cool, from a just basic, it sort of nails down, in my opinion, how these texture vibrations are represented in primary metasensory cortex, but also kind of highlights this, it's like rate versus temporal coding thing. This same population of neurons simultaneously are encoding frequency and amplitude in a temporal and a rate code, respectively. So you have these two kinds of codes that are coexisting in this one population of neurons. So again, though, we've proven, we've suggested, we've demonstrated that the information about frequency isn't conveyed in these temporal patterns, but we wanted to demonstrate that, again, kind of create that link with perception. Because again, maybe there's another part of the brain that we didn't record from that is a rate-based code, and that it could underlying this, could convey this information about frequency. So we did a perceptual experiment. We did a variety of them, but one of them was this one, where we had subjects. So we present subjects with pairs of vibrations, and we had subjects rate the dissimilarity of these vibrations. So if they felt exactly the same, you'd give them a 0. If one pair felt twice as different as another pair, they'd give it a number that's twice as large, etc. So first of all, let me just say in parentheses that this sounds like a really fluffy-duffy, like, well, they're coming up with numbers, right? But it turns out that if you have a bunch of subjects do this, there is a huge agreement between subjects as to which vibrations feel the same and which vibrations feel different. Okay, so then we have these dissimilarity measures, basically, that are gauging the differences of these vibrations along whatever relevant perceptual dimensions, in this case, probably spectral composition and amplitude. And then we want to see, can we predict the perceived dissimilarity based on firing rates alone? Like, basically, are vibrations going to feel dissimilar to the extent that they evoke dissimilar firing rates in cortex? And the answer is, we can explain 41% of the variance, right? And so that's not bad. It's not great, but it's not bad. And it's exactly what we'd predict, because to the extent that vibrations have different amplitudes, they're going to feel differentially intense, and that's going to be reflected in the firing rate. So about 40% of that variance is what we would expect. Now, if we try to predict these perceived dissimilarity or judgments based on firing rates and spike timing, so we did this in two ways. One, we just look at inter-spike interval histograms, and the other one we did using power spectra of the neural responses. Both ways yielded pretty much the same result, that we could explain substantially more variance in these dissimilarity judgments when both firing rate and timing was taken into consideration. So not only does the timing of the responses in somatosensory cortex convey information about the frequency composition of the stimulus, but it also seems to shape the way vibrations are perceived, and therefore, by extension, texture. So here's the big picture. I'm unbelievably on time, and I'm talking really fast, so I apologize for that. So the idea is that in the perception of natural textures, you have two different types of mechanisms. First of all, importantly, all three afferents can contribute to texture perception. On the one hand, you have like the coarse features result in these sort of these coarse skin deformations that are encoded in a spatial pattern of activation and slowly adapting type 1 afferents, are analyzed in cortex using these spatial filters that Jim and we have identified. And then on the other hand, fine textures, first of all, another thing that had been demonstrated that I forgot to mention is that in order to perceive the fine features of a texture, you have to move your finger across it, obviously. Otherwise, there's no vibrations, there's no timing, and you have no information about them. So what you do is you run your fingers across them. Vibrations are produced in the skin, texture specific. These vibrations culminate in temporal patterns of activation in PCs and RAs that are not only highly informative, but also shape perception. These are then result in temporal patterns of activation in primary somatosensory cortex, and somehow these spatial and temporal representations have to be combined to achieve a unified perceptive texture. And this is a question that we're actively pursuing. You know, when we touch the texture, we have this one percept of the texture as a whole, and yet there's these different mechanism at the periphery, different populations of neurons maybe in cortex. So the idea, so what we're trying to get at is how they're combined. So let me acknowledge the people who do all the work. Hannes did a lot of the analyses of the peripheral data. Let's see. Michael collected the cortical data, and Ali, who was an undergrad in the lab at the time, like a super undergrad, collected all the peripheral neurophysiological data and got a first author PNAS paper. Some people are just awesome. And these are my funding sources. Thanks for your attention.
Thank you very much. Very interesting. Any question or comment?
You're not going to ask me the question about 800 hertz again, are you?
I've got a new question for you. So I'm sitting here thinking about a part of the Braille dot story that you didn't talk about, that is distinguishing spatial variation from temporal variation, and
yes.
And that really is spatial. If you've got patterns that have different frequencies in the scanning direction versus the non-scanning direction, and that made me think about, for this fine texture, I wonder, in an ecological sense, and I'm asking for speculation, I guess, whether, in a sense, things are a little more two-dimensional, a little more spatial than just scanning in one direction. So if denim is a great example, or gabardine is an example, where in one direction you've got one periodicity, in another direction you've got a very different kind of texture. And I would think that ecologically the way that you distinguish those is to scan in different directions, and in a sense get some spatial information across time, you know, by, yeah.
So first of all, that's pretty cool that you said gabardine. I'm kind of proud of you right now. So
I meant to say corduroy, actually. And it was a word substitute.
All right, take that back. I'm not that proud anymore then. So, you know, the temporal code is definitely transforming space into time, right? There's no question about that ultimately what you're trying to transduce is a spatial property of the stimulus. Now, And there's also, it's true also that if you touch corduroy this way or this way, it'll feel different. And the way it feels different, we can predict based on the coding mechanism, right? Does that answer your question?
Yeah, right. I mean, I was just sort of bringing it up almost as a comment, and
Right.
I think you agree with me that in a real ecological sense of how people interact with fabrics,
Right.
you probably use multi-directional.
That's true. I want to also bring up another point. God, it just keeps slipping out of my head. Oh yeah, right. The key difference between a spatial code and a temporal one is that a spatial code relies on the relative activation of multiple afferents, whereas a temporal one is basically the computation is done on afferent by afferent basis. And I think the key is that these temporal codes, given the relative distance between afferents, could not be computed spatially.
So, was everything, were all of the textures periodic? So were they all textiles? Or do you also include non-periodic things like sandpaper?
No, I included non-periodic things like sandpaper. And actually, I have a bunch of, here, let me show you.
So, I mean, so
Yeah.
the thing that I was missing is, wasn't quite a lot of the argument built around the detection of periodicity?
Right, okay, so that's a great point. It actually, right. When you're looking at power spectra, it's nice when there's periodicity, right?
Yeah,
So that's why I show you examples where there's periodic structure. But in fact, all these principles apply even to the sandpapers.
Right. So it's
So
not
if
just,
you
you're
wanna
not just finding a single spike in the spectrum. You're talking about
No.
yeah.
No, we use the periodicity in the responses as a tool to show the relationships between these various things. But in fact, no, it's not a detection of periodicity. In fact, and one of the points that I sort of try to make is that, you know, periodicity is non-natural for the most part, right? Most of these, the textures that you would experience, I shouldn't say most, but a good number of them are non-periodic, like fleece, you know, the obvious one is sandpaper, but fleece, my brain is fried, but things like fleece. There's a bunch of them.
So just is it also possible to get large temporal vibration changes that are not due to roughness? So I'm imagining situations, so for example with glass actually has quite high friction if it's dry.
Yes.
And your finger kind of slides across it, right? So you're gonna, so I don't know if that's on the right temporal or spatial scale for that also to be triggering these kinds of mechanisms, but I'm imagining situations where you may get this structure, but which is not interpreted as roughness.
Right, so that's a great point, and that's true. It definitely, sticky things can create the skittering of the skin across them, but that skittering is accompanied, there's a fourth population of afferents that I didn't mention today because they don't exist in rhesus macaques, so we can't really study them, unfortunately, but slowly adapting type 2 afferents respond to skin stretch and are thought to be involved in so, you know, the co-activation of these, because this is a lateral sort of force, and those afferents are exquisitely sensitive to that, and then the brain learns to interpret that as differently.
So I'm trying to just sort of digest this all in my knowledge of what I thought Ken and Ed had done. Maybe you can just give me the big picture. So the simple story was that when you have subjective magnitude judgments of roughness, which as you said is a dominant component of texture, it's not all of texture, but you just ask subjects to judge it, they're going to give you very reliable things, and I'm sure on your textures would as well, and you probably measured that. And what was beautiful about the story is that within the domain that Ken had tested, that you could predict that you could predict it perfectly across the essays as a spatial variation code. Yes. And that's not saying that has to be true, but now that piece of data, if you want to expand the domain as you've done, you'd like to then, you know, can you replace that with an alternative code? It didn't seem to the end that you got to, I mean, you were jumping around between texture discrimination and distances of dissimilarity, but you never came back to like subjective magnitude over all textures. Did you ever?
I did.
Oh, maybe I missed that slide.
Here, I mean, if I understand you correctly, this is the slide.
Okay, so this
is perceived roughness as a function of, you know, so the top left one is an absolute exact reproduction. And so what you see, what I forgot to mention, you know, is a couple of interesting things that so first of all, look at all these textures that vary about 7-fold in perceived roughness and basically do not evoke any kind of response in slowly adapting type 1 afferents. And here's a thing. Here's a really weird thing. Essays, you know, if I say, what is the roughest thing, you know, it's going to be sandpapers. Essays don't respond to sandpapers. And yet they're the roughest thing that you can think of, right? So
I'm not trying to push this essay story. I'm trying to just see what the news story is. So
the news story is down here, right? The news story says you combine the spatial variation in SA1 afferents with the temporal variation in PC and in RA and PC afferents, and you can predict the perceived roughness of these textures.
So that down there is all your textures on the lower right, everything.
That is everything. That's right.
So that's now a weighted sum of different codes, if you will, across these different afferents.
That's right.
Which, you know, Ken didn't pursue this so far. You sort of alluded to it in my work. The idea that those codes have to be converted to an intensive code at some point.
Yes,
you know,
absolutely.
But you couldn't find that yet in 3B, is my understanding.
That is exactly right.
So you think that somebody somewhere might be computing an intensive code that you've approximated here. You just haven't found it yet.
Well, but you know what? You know what else you haven't found is an intensive code based on the slowly adapting type 1 afferent story, right? There was some modeling idea that maybe these receptive fields were. But you're absolutely right.
I'm not saying. I'm just trying to clarify.
No, man, I'm deeply offended now. I'm just kidding. You know, right? So no, I'm not saying we've figured everything out and like don't study texture perception because we know everything now. But what we know is that somewhere in the brain, because look. Bottom line is essays, there's no way essays are doing this. I mean, otherwise, sandpapers would feel very smooth. And therefore, it implicates these RAs and PCs. And therefore, we have to go look and find. But, you know, you bring up a really good point, is that when I in my my S1 story, you basically have a rote representation of the stimulus there, right? There's no elaboration of it. What I didn't say is that this is in 3B. In 1 and 2, basically, these neurons do not respond to vibrations. They don't care about vibrations. And so what we think, and we have some evidence, now we're actually recording the responses of S1 neurons to textures. These are vibrations. Vibrations are nonsense. Like, you don't need to analyze and elaborate vibrations. So what I think is going to happen is, and we have evidence to suggest that in areas 1 and 2, you present textures, and suddenly they respond very nicely to those textures. They're sort of filtering out nonsense vibrations, but if you excite them with meaningful, naturalistic textures, suddenly they become responsive. Does that sort of? But you're right. We haven't figured everything out.
I wasn't trying to say that, but I'll talk to you later.
All right. So this may be more similar to vision than you think.
Oh.
So ganglion cells have center surround structures, but surrounds are very diffuse and big and not very powerful. And without tiny eye movements, they don't give you edges. And so they also, with tiny eye movements, even when you're fixating, turn the spatial code into a temporal code, just like this.
Right.
And also, it's what? Oh, yes. It's bursts at edges.
Right. So I just want to. I think you're right, and I've heard this criticism before, and it makes sense that timing matters and whatever.
It's just
but
extra information.
The difference is that you know, in audition as in touch, you're extracting information from temporal from oscillations of a sensory epithelium, right? And so there's something kind of analogous like that in vision, but I think it's, I think if you're going to draw an analogy, it's more natural with audition than it is with vision.
Yeah,
it seems to me.
We don't have a scanning eye like, stomatopods or something like that, but it is true that if we don't have tiny eye movements, we don't
right.
have edge. So, but looking at this, so if I were to sort of look at this and think about, again, think about ganglion cells and parasol cells and midget cells, and I'd say, so your SAs, and if this was, say, luminance going up instead of roughness, that your SAs are like parasol cells. They have a very high gain early, and it saturates. So they're very useful for discriminating very close to the bottom. But if you want to code things up, then you have these cells, so especially the the PCs are actually, you have the straight line going up, but in the bottom they're actually quite flat, and so there's a nice big gradual increase in response with the roughness going up.
Except, right, but actually it's the PCs. I think if you were going to make that analogy, I would make exactly the reverse one.
Yeah,
it's the
I
PCs
think
that saturate, and the SAs actually are pretty linear over wide range, and not very sensitive at the low. PCs are the only ones that respond down in the sub-micron range, whereas the others respond,
you're right.
tens or 50
you
microns.
plotted it in the opposite way that we do normally. So we put the luminance on the bottom, and the spatial and the response.
All right. Well, sorry.
that was
it's too late now. It's published. I can't go back and. Yeah, bright. It's exactly. The abscissa is a neural measure, and the ordinate is a perceptual one. So I think this makes sense.
Right. There's no context. Luminance and brightness are pretty close and monotonically related.
All right, I got nothing on that one.
I just wonder, what's the minimum information necessary to discriminate the roughness from vibration?
What do you mean roughness from vibration?
So actually you can, even though you simulate the roughness kind of vibration, still it is possible that vibration rather than roughness. So
right. So these vibrations, again, to go back to this point I was making earlier about how areas 1 and 2 shut down. They don't respond to vibrations very well. Just like V2 and V4 don't respond to just mumbo jumbo visual stimuli, right? They only respond to things that, like, they don't respond to white noise, for instance, right? Kind of like vibrations like white noise to the somatosensory system. There is a fundamental difference, though, so the point is that vibrations don't feel like textures. There's a texture-like quality to them, but they don't feel straight up like textures. Now, roughness, let's say they did, right? Roughness and intensity would be different nonetheless, because we have another study that I didn't talk about, which showed that if you want to predict the perceived magnitude of skin vibrations, that's a rate code, just straight up, sum all three, and you can, we get 97% of the variance explained in magnitude judgment. So it's a different computation that's being affected. This is a variation metric, whereas magnitude is just a straight up rate metric. So they're different quantities.
Okay. Any other question? Okay. Okay, thank you very much. Very interesting talk and exciting discussion. Thank you.